So I think I'm online again. Give me a few minutes, a few seconds to get everything sorted out. I think sound seems to be okay. Okay, I think we're ready to go. Uh, Hello and welcome again. Microbehunter here. I'm Oliver, and today, yes, I'm going to put some blood under the microscope. It is a special request live stream. Yes, school just started. You might know that I'm a teacher teaching biology. I asked my students, "What do you want me to put under the microscope during the live stream?" And several students said, "Well, we want to see some blood cells." I said, "Yeah, that's possible. So let's put some blood under the microscope today." Some students wanted to see some white blood cells. I hope that we're going to be lucky. And not only that, of course, I'll be also staining them. It's not the first time that I'm putting blood under the microscope, but every time it's a little bit different because, yeah, you cannot always have the same parameters everywhere. Well, just a short thing for those of you, if you're new to this format, the first 3 to 5 minutes or so, I usually say hi around the world so that essentially I give my viewers some time to join into the live stream. If you have any comments or questions, please feel free to post them in the chat. But please also indicate maybe at Oliver or at Microbehunter, so that essentially, yeah, I know that I can skip ahead and just yeah, look at those comments here. Okay. Yes, there are not only students I see that have joined here, but also some of my colleagues. So hello from Vietnam, from Grainsville, Florida, from Greece, England, from Austria, yeah, and Rhode Island, the United States, England, Poland, Lake Constance in Europe. Yeah, yeah. They just want to see a teacher in pain. It will be a little bit painful because I will have to prick myself today, right? Yeah, and also from the UK, from Austria, United States, very nice. Okay, so if you Northern Australia even, so I know that I've been asked why am I making the live stream at this particular time right now. My local time is 9:30 in the evening. Why that? Well, according to my time, why that late? The reason is it has personal reasons because I myself just returned home as well, and yeah, also I know that it's not always convenient the time for everyone around the world, but yeah, so we'll just basically stick to the time as it is. So what am I going to do today? Look what I got here: some lancets to get some blood, and I'm going to simply put some blood directly under the microscope, but also look, I've got some staining solutions here as well. Löffler, that's methylene blue solution. We can stain the white blood cells. And I've also got some salt, concentrated salt solution over here to have a look at some, yeah, a little bit of osmosis, plasmolysis in other words. So if we add salt solution to the blood, we should actually see the red blood cells shrink. Yeah, so this is actually something that I would like to also demonstrate. Now, I know that I've changed the format around a little bit last time, where I made 15-minute chapters. Today, it will be again a little bit more in a traditional way because I want to take my time a little bit because, yes, this video actually will also be used for instructional purposes in school. So for this reason, yeah, I'll take my time and just also show you how to put some blood under the microscope. This is alcohol because I will be, yeah, disinfecting myself a little bit. Yeah, so it's actually concentrated alcohol, but isopropyl alcohol also works, or 70% alcohol also works. But yeah, you always want to make sure that everything is nice and clean. Yeah, it's a pen. I know. It's there is this pen that you can use for people who have diabetes, for example, or who need to take a blood sample, where it's psychologically less, how do you say, straining because you don't actually see it. What you have to do, yeah, I didn't get those pens yet. These are special pens that are basically you can. It's much easier to prick yourself. So look, I got still several of those here. Those guys here, look. They're cool. So there's also a date on here. So how best best before date somewhere. Doesn't matter really. All medical products have to have those. And essentially they're sterile. I just wanted to talk a little bit also about some background info. How do you actually get them sterile? Basically bacteria free, because during the manufacturing process you cannot really get it free of bacteria. So what you do is you just make them in a normal way, and then you take the whole box and the whole pack and you just send it through a radiation. So basically it's going to be irradiated, and the radiation will actually kill all the bacteria. So it's sterilized, or basically after packaging. So that's how this actually works. Okay, so hello from Ontario. Okay, and what I'm gonna do now is we're just gonna take it from there, and this is the tough part. So if you cannot watch that, okay, then just look away. And again. Here, I think I'm just gonna take, for whatever reason, I prefer the middle finger, because I can use my thumb to press against it, and so this is gonna be the difficult part. I have to tell you, it's also not easy for me, right? So it's new, so I have to kind of twist it here, right? And here it is. This is the needle. This I can throw away. Right? You know what? You want to have it nice and big, right? Here we go. Yeah. Ooh, this is difficult, right? And what you do is sometimes what helps is you might just basically hold it like this, right? Yeah. And then you can actually also flick it a little bit like this, because then you don't have to watch it. Yeah, like this. Okay. Then it's less psychologically a problem than having to actually see the needle go into your skin. Yeah. So I'll be using it again later, so I'll keep it clean. And you press, obviously. Here it is, the blood. And what I do is apply a little drop of blood here just keep on pressing, and I just apply on here on the side. Like this, okay? You take a second slide. I'm going to show you several methods on how you can do that, right? And in the lab, what they usually do is they take a second slide. Yeah, they go like this. You see how the blood spreads, and then you just swipe over it. Okay? And this will give you a very, very thin film of blood. Yeah? This is, of course, something that has to be put away. I'll clean myself here again. And what you can do now is you can actually air dry it, and you can stain it. Staining, there is this method called methanol staining and so on. Well, I'm probably not going to do that because I don't have that. But we're just going to, you can actually see how it's actually drying. And we're just going to put it directly under the microscope without a cover glass as it is. Okay, and then later on I'm going to show you also how to a couple of different methods, but I hope that the layer is thin enough so that we're able to see the individual blood cells. So I need to now switch over to the microscope view. Here we go, and I need to go up, and with the brightness a little, oh, this is dark field, unfortunately. Okay, here we go, and you can actually see what I'm doing here on the top. And I close the condenser, I flip out the whole thing, and so this is now bright field. Okay, and no drinking in the lab. I just returned home and I was still a bit thirsty. So that's basically how it looks like. Let's have a look. This is still very dense. Okay, very, very dense. So over here maybe you're gonna see where are the cells here, right? Yeah. So basically, chemical. What I heard is some people actually use alcohol as well, methanol. Yeah. But I've never tried that because methanol fixing. I don't wanna play around with methanol. So that's basically now. The 10 times. I'm going to. Here we go. It's 20 times objective, and now we are able to see those round circles here. These are basically the red blood cells, and you know what? I'm gonna put it in DIC. Some people were asking me why does the blood not look red? Yeah, that's because they are so small. And here 40 times, and here we are, right? And here we already have the red blood cells, and 60 times. Yeah? In case you're wondering why it looks kind of blue, that's because of the optics that I'm using here. So I can change the color around. Yeah. And what you can then do, of course, is you can admire a little bit the, how do you say, the shape of the red blood cells. And another thing I want to show you is the following. Let's go down and let's have a look how it looks in phase contrast. That's also a possibility. Okay. I have to open it. Yeah, this is how it looks in. I have to open it a little bit. This is how it looks in phase contrast. Also quite nice. Okay, so and the idea that I want to basically do today is I'm going to then later also, of course, I wanted to try some staining as well. But let's just have a look at the whole thing here. Yeah, using. Yeah, and one of the nice things that we see here is that the red blood cells, they are forming a single layer, right? Because I spread them apart across the whole slide. Look, and things like this here, these look a little bit like, how do you say, white blood cells here? But I'm not quite sure. I'd have to stain it first. Yeah. So, and there's a Lysham stain highlights the nucleus of the white blood cells. Yes, I'm going to use methylene blue, and you can actually also see the nucleus of the white blood cell. Yeah. So what I don't have it anymore, but there is also something called a hemocytometer, which allows you to actually count the number of red blood cells, that you have per milliliter. Yeah, but what we have here is that this one is already a sample. It already is basically dried up, right? So they are kind of sticking to the slide, yeah, and are therefore not moving. But when I show it to you with a cover glass, then we can actually see them move around on the slide, and you can actually then also see how flexible they are. Yeah, so that's basically how it looks like. What I have not tried yet is usually when I stain them, I do it slightly differently, but I just want to try it out by putting a little bit of stain directly on this blood smear. Let me check again the desk. Here it is. Okay, and with a cover glass, and then we're going to see if this actually results in the white blood cells to be stained. Let's have a look. And I'm going to now use Löffler's solution, methylene blue. And we're going to take a very, very small drop. Yeah. And I hope that this is... I don't know, this is not very small, but... Drop the cover glass on top. You know what I might try? I have another idea. I'm a little bit worried that it might be too much stain here. No, I'll leave it like this. So, yeah. And any excess, I'm just going to use a tissue paper and I'm going to remove that. Capillary action at work. Okay. Now, in the Löffler solution, there is also potassium hydroxide, KOH, and alcohol. So, yeah, this might actually negatively impact the red blood cells, but it should stain the white blood cells. Okay. Okay, I'm occasionally reading your comments as well. Okay, so every time when I'm pausing a little bit. So, and let's have a look how this actually looks like now. And in this case, of course, I want to put a different filter in here. So, where is the scope view? Okay, I've lost the focus, of course. This is how it looks like. Okay, let's go up a little bit with the. I have to go up with the brightness a little bit here. Okay, we see the contrast is much lower now. Okay. And what we can do is we can have a look now. And this is already illustrates something very nice. So I'm using number 60 times objective. I do have an arrow over here. So what you see over here, these are the red blood cells, right? Yeah. You see they have lost a little bit their shape, and that's because of the because there's some alcohol in there. So it could be that some of these white blood cells actually, red blood cells actually popped open. But look at this here, this guy in here. Yeah. This here is a white blood cell. Yeah, and you see that there's this darker, I don't know, round, half round thing in here. That's the nucleus. And you see that the nucleus of this white blood cell is irregular and it's lobed. Therefore, it's called polymorph nucleated. Polymorph means many shapes, right? So that's there's some different categories of white blood cells like polymorph, nucleated, granulocytes, and whatnot, right? So it is possible to have a, how do you say, to identify the different types of white blood cells by looking at the shape of this nucleus. Yeah. So this is, usually you wouldn't use a cover slip if you have fixed the cells and stained them. Yes, that's correct. But essentially they're not fixed. I basically just spread them apart, and I simply dropped the stain on it. Yeah. So and then you can here, I don't know, maybe not, but just want to see show this actually also illustrates a little bit that the white blood cells are much less in how do you say, in concentration in the red blood cells. Yeah. So I have to see. Here's another one. Okay. You see that the nucleus is actually has several parts, and they're actually connected. If you zoom in yet more, you can actually see that the different lobes sometimes are actually connected by. So that's another white blood cell. Why does Löffler stain the red cells as they do not possess a nucleus? First of all, that's correct. Löffler solution methylene blue, basically, well, red blood cells do not have a nucleus, right? And you can actually see that over here, these are the red blood cells, and they're barely stained. So those dots that you have here are probably not the nucleus, but some other things, maybe proteins or whatever, right? But you see actually that if it actually if a nucleus is present, right, then it will actually stain it quite well because the Löffler solution is alkaline, it's basic, and the DNA, which is in the nucleus, is acidic. And therefore, the basic alkaline stain will react well with the DNA. That's why it stains it well. Okay, so that's the reason, and essentially what you can do now is you can have a look and you can count and identify the different white blood cells that there are. What I once have done, and that's actually wow, this was pretty. I had a tiny little infection here, a pimple, and I squeezed it out, right, and the pus, and I also stained it with methylene blue. It was just 100% just white blood cells, all over, just no red blood cells. So it looked quite different. So essentially, what we have to do now is we just have to go through the whole thing here. You know what? Maybe at a lower magnification is actually better because we have a wider field of a view. Here's another one. Okay, another white blood cell. So so you see it's a fairly easy thing to do. But what I would like to try as well is I would like to try also a different stain, and I have to admit I have not tried the other stain yet. Okay, it's crystal violet. It's a very powerful stain as well, and I would like to also try that. And honestly, I really don't know how well this actually works. Again, the cells are not fixed to the slide. I wonder if heat fixing actually works or not. I've not tried it, right? But let's try now some crystal violet. This one is actually very, very powerful. And this means that I have to remove the stain again. Okay, because otherwise I'm not able to see anything at all because it's totally blue. So what I'm going to do therefore is first of all have another sip of my coffee. And give it a minute or so. And I'm going to remove again the stain. And I'm going to maybe wash it a little bit to remove any excess stain as well. So we're just going to do it like this. I'm going to discard it. So here we go. I don't want to scratch the surface too much. So, and here I have got some water. just gonna use this pipette here. And I have to be a little bit careful here because, of course, I don't want to rinse away any cells, which is probably going to happen anyway, right? Or let's try it like this. Okay. And a cover glass goes again on top. And this is now a little test for using crystal violet. Yeah, actually, see, there is still plenty here. Let's have a look. I really have no idea how well this works, okay? That's one of the things that I also like to do every now and then. It's simply some trial and error. I do also want to switch over to the scope view. Yeah. Can we use the crystal violet solution in class? Theoretically, yes. I mean, I don't know if you are one of my students. We actually have a whole bottle of crystal violet solution in school. The only problem with that solution is it's extremely strong. So I just want to show you. Actually, it looks kind of nice what I have here. I want to actually show you. Once you've got crystal violet somewhere, I'm going to show you the desk here, my desk. Yeah, it's very difficult to get it out again. So that's why this is kind of a plastic surface that I've got here. So that's a little bit one of the worries that I have is once you actually got it on the table, very difficult to remove, if not impossible, even with alcohol. Yeah. So I want to see now, what in the world is this even? Wow. Look at this, 40 times. Look at this, look at this, look at this. Yeah, these seem to be the red blood cells, which are stained blue now. So you see the different stain reacts quite differently. And those large things here, maybe these are white blood cells. Yeah. But you see that they are kind of a little bit deformed as well, which is not surprising because if the osmotic balance is not right, yeah, the blood cells tend to shrink or to swell and pop. Yeah, but I think I think the idea that look here. Here, do you see this here? This seems to be here a white blood cell. Yeah, with the nucleus here. It seems to be a little bit popped open, but I think you might get the idea that this stain looks quite different. Yeah. Looks like the crystal violet concentration was a bit too high. Yep, that could be. However, I have to say one thing. The fundamental problem, even if I were to dilute it down a little bit, because the crystal violet reacts with the cells will actually soak up, so to say, and react with whatever stain is there and still might become very blue, right? So that's a little bit the issue. It is, of course, it's a question of concentration. You see a little bit of stuff floating around, but the cells react with the stain. So even if it is dilute, it can be that the reaction is so strong that they will turn quite dark. Yeah. So yeah. So let's have a look here. Yeah. So these are here is another white blood cell, but you see that even the cytoplasm of the white blood cell is stained so blue that we cannot see the lobe nucleus here. Yeah. Nile red I did not try because I don't have it, unfortunately. So Nile red is also a stain. Yeah. So let's compare this again a little bit, okay? Just for comparison. So this is now using crystal violet, and this one is how it looks like when using methylene blue. Yeah. And you see that most of the red blood cells are not stained, but we're able to see the white blood cells much better here and here, and here. And you can also see that those white blood cells here, actually it's a good one because there are 3 of them quite close together. You can see that those 3 white blood cells actually have a differently shaped nucleus, and that's how those folks are able to distinguish them and categorize them. Yeah, so you have a look here. Yeah, you have a lobed nucleus here, 4 of them. Yeah, and here's another one. And here, I don't know where the third one, the fourth one is. Yeah. But So that is this was the methylene blue, and this one is the crystal violet. And you see that the crystal violet is pretty aggressive. Just stains everything. Yeah. Yeah. So. Yeah, so that's this was kind of the So the methylene blue is more efficient for locating white blood cells compared to the crystal violet. I would probably say so, even though this methylene blue is not the most common stain to use for blood. There are specific blood staining stains as well. And this one over here, look how the red blood cells actually how they they look completely deformed here. Yeah. So if you don't, why do you mix the water and the color on the glass and not separately? This is was one of the questions here. The reason is it's simply for practical purposes. I do not want to dilute the original crystal violet down, right? I just want to keep this concentrated. And of course, what I could have done is I could have taken a drop here and mixed it separately and diluted it separately. Yeah, that's definitely possible as well. Yeah, it's a little bit of convenience. I just mixed it directly on the slide. Yeah. So I'm just reading again some of your comments here. Yeah, can we use the crystal violet in class? I'm saying, yeah, well, you could, but I'm a little bit worried because if anything is spilled, it's impossible to remove. Yeah. So that's a little bit the thing. So you know what? I'm checking the time. It's a half an hour now. We can actually, I don't know, try another one. If you want to actually see, how do you say, something else, then we can actually. I mean, I did also prepare some. How do you call this? Some salt water. We can do this as well. Let's try something else. I mean, I do also have other stains over here. This I have. I never tried those. You know what? Let's just try them. Saffronin stain. It's the red one. Yeah. Then I have got acetocarmine. It's also another red one. I've got iodine and I've got regular fountain pen ink. You know what? Why not try it? I mean, I think it's Basically, I'm gonna rename this video trying out different stains on blood. Yeah. So we've now looked at methylene blue, Löffler's formulation, and we've looked at crystal violet. So let's do another one and let's try now some red stains. I have to be honest, not knowing how they look like. Okay. So if you're new to this format here, I like to sometimes simply try out things that I've never tried out before, and sometimes we get some interesting results, and sometimes not. Fuchsine. Oh, that's fuchsine, aqueous. That's red. Saffronin is red, and acetocarmine is also red. So these are 3 red stains. Ooh, I have to prick myself again. So I'm going to put the tip away. Okay. I'm going to take another slide. Where's my slide? Slide box. Just a second. Here we go. Here is another slide. Okay. Ooh. Yeah. Let's see if this. Am I still able to get some blood out? Nope. Unfortunately not. I will simply try it again. No, I'm going to first quickly, briefly disinfect it again. Just you can see there's this little red dot from before. So we'll try the same. Here we go. Here we go again. That was a lot. Let's try it again. This was, okay, I'm going to clean, spilled a little bit of blood around. So let's give it another try, like here. Allow it to spread, and then here we go. Okay. And now I'm going to try, you know what, I'm going to air dry it again a little bit. You can actually see how it slowly starts to dry. And I'm not going to heat fix it because I've seen that actually it also works, yeah, as it is. What does heat fixing mean? If you watch one of my previous videos, usually bacterial suspension, when you've got bacteria, you can actually put the whole slide over a fire, a flame, and then after you've dried it, and then the bacteria will stick to the glass slide. Right? Yeah, and I'm not going to do that here. Yeah, I can actually try it as well, but we've seen that this also works like this. So next thing is, yeah, I'm going to now use, again, 2 cover glasses. And I'm just going to make two, I don't know, the first one is safranin. It's, yeah, aqueous means it's water-based. I don't know. Really, I really don't know. It's kind of red. Here we go. And let's drop this on here. Way too much again. The question here is, do you buy stains as solutions or mix yourself from pure stains? Well, both, as a matter of fact. So, for example, fuchsine and safranin, I basically bought in powdered form from a microscopy supplies company, a British supplies company. It's called Brunel Microscopes. I'm not affiliated with them, so yeah. And they basically you order it, and you order it in powdered form because they are not allowed to send. There are certain rules. You're not allowed to send liquids, yeah, for whatever safety reasons. And for this reason, many stains are sold also in powdered form. Okay, and this is yeah. When I mixed them myself, and some of the stains from chemical supplies companies within the country, like for example the crystal violet, already came in liquid form. Yeah, so this one was what? Yeah, safranin or fuchsine? I forgot. No, it was the safranin. So let's have a look now. And again, I have no idea how this actually looks like because it's the first time that I'm staining blood with that. So let's have the scope view again. Ooh, okay. Let's go up with the magnification. Ah, look. The red blood cells. Yeah, you can actually kind of very dense here. Yeah, you see they're very dense. They're sticking together, right? But I wonder if we're able to find any white blood cells here. Yeah. Maybe this one over here, in the center. Yep. Also seems to work. Okay, but I think the blue one is actually a little bit nicer. Yeah. The way you prick yourself with no hesitation is crazy. You get used to it. It's not the first time that I'm doing that. You do get used to it. But as said, it's as soon as you start thinking, yeah, then of course you see. This one is a nice example. The density is for whatever reason is too high. Yeah, so you see that the red blood cells are all sticking together here, right? Here's another one. Another white blood cell, also not quite nicely visible, but you see over here again the nucleus, which has the different lobes, the polymorph nucleated cells here. Yeah, so that's safranin. And what about, I don't know what this is. This could be some kind of junk maybe. Yeah. How do I view a live blood smear? We're going to have a look at a live blood smear as well. Okay, don't worry. I'm going to show you. Ideally with a cover glass, because if you do not use a cover glass, then it's going to dry out too quickly. Yeah. So again, 2 of them here. Yeah. If you, we're going to see this. If you dilute it with water, the blood cells are going to pop. You're not going to see anything. Yeah. So no water, please. Yeah. So that's yeah. Yeah. So that's one of them. Let's try another one. Another stain. So this was what? This was safranin. Now let's try fuchsine. The desk view. So that's another one. Just because, I mean, I don't have to tell you that, right? Just because stains have the same color does not mean that they behave the same way chemically. Obviously not, right? Yeah, so we can actually also try to do the following. We can actually remove directly some of this stain as well. Yeah, and the rest will spread like this. Okay. Yeah, it's a different type of red. Okay. Okay. Yeah, it's you can dilute it with saline, which is basically salt water. Yeah, to prevent this from happening, to prevent the cells from bursting. So we have now again the scope view. Ooh, not a nice one. Okay, here. Okay. Ah, yeah, here maybe. There is I don't know. There's lots of junk here. Maybe it's you know why? Do you know why there's so much junk here? Because it is the right side of the slide where where there was a lot of blood, so the density seems to be too high. It's an air bubble. See where the air bubble is? You can actually see the red blood cells, the individual cells. Yeah. Oil immersion. Let's do that later. Okay. I cannot do oil immersion here yet. I mean, I could, but I wanna actually see it nicely. Yeah. See, that is not a how do you say, a nice example. Here you can see the cells a little bit better. I wonder if I'm also able to find some white blood cells here. Again, the red blood cells difficult to see here, but these are the tiny circles here. Okay, that's seems to be a little bit coagulated blood. Yeah, so let's have a look. So let's have a look here. No, let's go up a little bit with magnification yet further. Yeah. Not so yeah, not so good maybe. Yeah, too dense. So we see that there is maybe also a little bit of trial and error needed to also get the concentration down. Yeah. But you see that the whole thing is liquid and it's flowing, right? Yeah. Yeah. I don't speak English, but it's still interesting to watch. What you can do is there is an automatic translation function. Okay, so this means that afterwards the video will be online and you can watch it online, and then you are able to also change the language around. Yeah, there's a thin looking film between all of the cells with the S. I noticed that. Let's put it between somewhere between the cells here, right? There seems to be some kind of a I don't know, a film, and I wonder if this is some kind of the protein. Yeah, of the blood plasma. Yeah, indeed, that is also stained. You can actually see it over here quite well, right? You see that there seems to be, yeah. So it seems to be that indeed it seems to have stained the blood plasma. Yeah, fibrin could be. Fibrin is the protein that actually when it clots, yeah, or fibrinogen, which is the precursor protein. Yeah, so you can actually see also here that. Indeed, it seems to have stained the plasma a little bit. So again, for comparison, this was the fuchsin, and over here, this here is the safranin. Again, very different. Yeah, and yeah, as I said before, is this the type of chemical actually has an impact on, yeah, also on the shape of the cells. Yes. So basically because of the osmotic balance. So, what are the red blobs around the slide? Those big red blobs. It's a good question, and I think it could be some kind of yeah. This is at the part of the slide where the blood concentration was high. So either some kind of maybe coagulated blood, yeah. So, but this is because when blood coagulates and the red blood cells stick together, you cannot see them as separate cells anymore. Yeah. So this is actually something I would say these are artifacts. So they shouldn't, yeah, those blobs, yeah, probably shouldn't be there. Yeah. Wout, researched something here. Apparently, fuchsine stains polysaccharides. Just looked it up, so I guess it's possible that it stains the plasma, the glycoproteins in the plasma. Yeah. Yeah. So it's quite interesting. Yeah. I think at least it's interesting. It's a little bit of how do you say, yeah, hands-on exploration. So what I'm on, let's go back to the desk view. Okay. Yeah. So we now tried two blue stains and two red stains. I don't know, what we could do is we could really go up with the magnification a lot, and we could try to look at some not dry red blood cells. Yeah, so when they're still a little bit liquid. And here I just want to show you how I'm doing that. Because we need to get them still very thin, okay? So my suggestion is, if I've tried this before and it worked quite well, you take again a new slide, and what you do now is you do not take the small cover glasses, but look what I got here. I've got those, I'll quickly put the stains away because I need a little more space here on my desk. I've got those huge, gigantic cover glasses here. Look how big they are. They're almost the size of a slide, right? And this basically means that when I put a drop of blood under the cover glass, it's going to spread beneath the cover glass into a very thin layer, obviously. But because there is the cover glass on top, it does not evaporate so quickly. And that's actually pretty good, because then it stays liquid, and then we can actually see, when we really zoom in a lot, that the red blood cells are actually quite flexible. Right, so this is something that I want to show you now as well. Look, I just realized that this cover glass here has it's cracked. I cracked it. There was a small crack already in here, so I'm gonna put this away somewhere. Let's take a new one. So what does this mean? This means I have to I have to prick myself again. Okay, so here let's do it again. Yeah, you see, it heals so quickly, yeah, because, yeah. So, number 3. Maybe hold it like this and then go like this, and then, yeah. It looks pretty scary. Sometimes it helps to get out more blood by twisting it a little bit. This does hurt a little bit. There are lots of, how do you say, nerve endings in the fingertips, right? So what I do now is now I will put a drop of blood on here. It can be a little bit more even. Up. Because I want to have a little bit more blood, because I want to keep it liquid. Okay, so, and now the cover glass, and look what I'm doing right now. I'm now going to put the cover glass on here. I'm going to drop it directly on here, and we're going to see how the blood spreads across the slide. And this is what I would like to have a look under the microscope, okay? As the blood spreads. Ooh, there is a, how do you call this? Here we go. There's a glass fragment from before. So let me quickly get the scope view. It almost spread completely already across. Okay, so here you see they're moving red blood cells as they spread by capillary action. They're so moving so quickly that you cannot even see them. What a pity. Here we go. Yeah, that's okay. Again, I gotta do this again. The reason is because, okay, here it's a little bit thinner. Yeah, here maybe you can see it better. There are too many of them, right? But you actually can see how they spread. So I probably have to do this again with, how do you say, less blood. Okay, but you get a little bit the idea of how flexible everything is here, how flexible they are. Let's zoom in a little bit more. And because it's so thin, the layer, yeah, they are also pressed flat a little bit. What about here? Let's have a look here. Here, that's exactly a little bit here, just slow down here. Yeah. Yeah, so I used too much blood. Okay, so the density is way too high, so I'm probably gonna try it again. Yeah, it's way too high, the density. So we need to, I don't know, way much less blood. Huh? And then we can also use, how do you call this, oil immersion, because then we can really magnify them nicely. So somebody's asked, since we're starting with cells for the high level course, are we going to be doing similar experiments to this, to staining blood, or maybe looking at cheek cells again? I'll be honest with you, no, we're not going to be looking at blood. You know why not? In school, because it's not allowed. Yeah, there are actually laws, rules that prohibit basically doing experiments directly with blood for hygienic reasons, because otherwise maybe diseases can spread. Back in the day it was HIV/AIDS, obviously, right? But hepatitis and so on and so on and so on. So actually, working with blood in human blood actually is not allowed. Okay, so they've adapted to the laws. And that's actually also one of the reasons why I'm making those videos here, because sometimes actually it's easier to simply show the things using my admittedly very nice microscope. Sometimes you actually see the things better, right? It's easier kind of to observe them. So this is actually a nice thing here. What you see here is what you call rouleau formation. Look, that some of the red blood cells start stacking up like a roll of coins. That's the French term is called a rouleau. And this is a normal thing that happens when blood starts to dry up. Okay? The fact that the red blood cells, they have a very large surface area on the side, and therefore they like to stick together, right? And that's a normal thing. Why do I keep on saying this over and over again, that this is actually a normal thing that you can observe? Because there are some folks out there who think that you're able to diagnose certain illnesses simply by looking at the shape of the red blood cells and at whether they form those stacks or not. This doesn't work. Okay, so this is So you know what? We're gonna start again, all over again, and with a little bit less blood. Okay. Because if we want to really see the cells, the individual cells quite nicely, then we have to make sure that there are not so many of them over each other. Look, I'm gonna go to the other end of the slide. Look, okay, so I'm right now on here, seen from my view. That's you can actually follow here at the top. That's on the right side. And now I'm going to go over to the left side where actually I put the blood drop, and also check what happens to the density of the cells. You see? Yeah. As I move over, it becomes more and more dense. Here it's still flowing a little bit. And here we're not able to see the individual cells anymore because they're so tightly clumped together, right, that we cannot see them. But it does, the thing why I still like this series is because of the movement. It kind of shows still it simulates a little bit of how the red blood cells actually try to move through blood vessels, even though there are no vessels here. Yeah. The students might mistake me for a pincushion. I once used to say, if you're suffering for the sake of science, so to say. Yeah. Yeah, but that's basically why they're moving is because on one edge of the slide there is lots of blood, there's little on the other, and by capillary action, the pressure of the cover glass, this pushes the blood along, right? And there are some places under the microscope slide, like over here, where the blood has already started maybe to clot or is very thick, so it's not moving. Yeah, and but it looks a little bit like a blood vessel, even though obviously there is no blood vessel here. Yeah. But still a nice demonstration, a little bit, also how many red blood cells there are. It's really crazy. I mean, you see, in biology lessons, the best thing that I can do is I can draw a red blood cell on the chalkboard. Yeah, but actually seeing this stuff with your own eyes directly on, live, so to say. Of course, I could have prepared videos and pictures, but it's actually something different if you actually see the whole process. You prick yourself, you put it on the slide, and you can actually see it directly. It's kind of how do you say? Even for me as a biologist, it's still fascinating. Yeah. It's just the enormous number of cells even, right? So you know what? Let's use less blood, and then there has been the request. There has been the request to use oil immersion, so that means go up to the maximum magnification possible. Yeah. Can't you find a blood supplied place on the human body with fewer nerves than the fingers? Yeah, ears. Ears. This is where doctors actually take blood samples as well. At least I've had them once taken. They measured some parameters in the hospital, and they've taken the blood from the ear because there are indeed less nerve cells in here. That's why it also doesn't hurt so much when you get some kind of piercings here and so on, right? But that's actually, yes, this would be the place. But honestly, it's not so practical to play around with my earlobe. Right. That's why I'm using the finger, and it's also easier for me to show the finger directly under the video. So you know what this means? Another one. Never pricked myself so often. So yeah, as a general rule, I always disinfect myself first. It's unlikely that but still. See, and look, it already stopped bleeding. Yeah. So let's prepare everything again here in another cover glass. And what I want to do now is I want to now really go up with the magnification using oil immersion microscopy. Okay, so then we're going to go to, that's the theoretically highest magnification that I can get. Yeah, I already stopped counting. You know what, who cares? It's just push normally, it's all that works. Turn a little bit, and this wasn't enough. That's too small. Another one. So, someone doesn't want to go in. Why not? Yes, here we go. A small amount, I said, right? So, a small amount. I think that's a little bit less, okay, than before. So, and yeah, let's give it another try. And let's carefully drop the cover glass on top here. Yes, I think that's, yeah. And what we have now is the following situation. You see that essentially it's not able to spread all across because there's very little. So that means we should be able to see individual cells here, right? So let's try to find that place. Where is this? So that's, you see, that was the other one. So I'm going to change again over to the scope view. So that is, yeah, this is the dense part. Moving, and let's go over to the less dense part. Ha! Yeah, here still moving. Let's see if we can. I forgot to put in the DIC. Right, and here we go. Here, that's what I wanted to show you. Here, these are now the individual cells. Yeah, and the space is so narrow between the slide and the cover glass that they're also pressed flat, right? But somehow they're still able to squeeze through. You know what I got here? Look, I 3D printed something. Just want to show you. Look what I 3D printed. This is, I don't know if you're able to see this. That's a red blood cell, right? And if the slide and the cover glass, right? Yeah, it's so narrow, then yeah, that's why they're pressed flat, huh? And that's what we see here. See, it's already stopped here. Slowly they're moving ahead. Let's go up with the magnification. It's a little bit blurry. I'm not so happy with this quality here. Let's try this here. Maybe that's better here. Yeah. Yeah, so you see they are now pressed flat quite a bit. And I promised you that we might actually You see, yeah, look. That's actually quite nice. Over here, they're kind of pressed flat, right? And over here where the layer is thicker, yeah, you see that they have a more, yeah, they're able to move better, but they also have a slightly more normal shape. Yeah, exactly. Yeah, so they're able to, yeah, that's actually better here because here they're not squeezed together so much. Yeah, and and there are a few other things that, you know what, let's go maximum magnification just for the fun of it, okay? So this is now, what I'm gonna do now is this oil immersion microscopy, and that's pretty advanced stuff. So I generally don't like it so much because it's a little bit messy, but what I have to do now is if you've never done this before, you can might watch a little bit of what I'm doing over here. Over here, I'm going to use my arrow. Do you see this objective over here, the one with the white ring here? That's my oil immersion objective, and I'm only allowed to do that using this objective. Otherwise, the others are gonna break. This one is a special objective. It's an oil immersion objective. And it says oil on it, which you cannot read, but it says oil on it. And what I'm doing right now is, I don't have Eocene available. Sorry, somebody asked. So what I'm doing right now is the following. I'm now going to put in the oil immersion objective. And I'm going to focus a little bit, and we just remember how it looks like. We see them, but it kind of looks a little bit, how do you say, not very bright and a bit blurry. I can try to, you know what, I'm trying to remove this, yeah. Yeah, so that is now theoretically the highest magnification, but what I'm doing right now is the following. I'm now going to, yeah, I've got this focus lock lever. I've got a slightly better microscope. So look what I'm doing right now. I'm lowering, so it's completely losing focus, okay? That's clear. I'm now going to put this directly on the slide. The oil. Okay, directly on the slide, and I'm going to now raise it again, and the objective is now going to be covered in oil. Of course, I have to clean it later on, and you see that the brightness and clarity is much better. Okay, so that is now, yeah. I'm going to put in the DIC as well. Let's open this up a little bit. We can add a little bit of color if we want to. Yeah. Yeah. So there was a question of, wait, how does that work? Is this question for me or yeah? What I have done right now is I have now basically there is now no air between the cover glass and the objective. It's covered in oil. Right? It's an oil-tight objective, and this gives the maximum resolution. It has to do something with a refractive index. Yeah? And what I am occasionally doing as well is sometimes, and you have to be really, and this is where, honestly, where we have to be a little bit, how do you say, very patient. Because sometimes, and unfortunately I didn't have time to prepare a video, but those things that you see over here sometimes, you might actually see them move around. Some of them are actually white blood cells that are moving. The reason why the red blood cells here are moving are again because of the capillary action. Okay, but some of these things here, over here, this one here might be a white blood cell with a nucleus in here. And some of them are actually moving around searching for bacteria. Yeah, and I should have actually prepared a time-lapse video. Yeah. Yeah, but that is essentially, I personally like this a lot because this really shows quite nicely how they look like and how flexible they are. Yeah. The red blood cells. Yeah. Can simply have a look at this, enjoy this for a little bit. Yeah, and yeah. Sometimes they get stuck a little bit because of the small space between the microscope slide and the cover glass, and then they have this droplet form because they kind of stick. Yeah. Something in the middle, this one over here, yeah, could be, and I don't know, it could be a white blood cell, right? Yeah. Can you tell us how you recorded the time lapse, the slow motion video of the white blood cell? What did you do to keep the blood from drying out? So what I've done I've made. It's actually in this channel or in this in the other channel that I have is this time lapse video. And what I've done is I looked at a place where the red blood cells are not moving anymore. So I gave it some minutes for everything to stabilize. And then I looked at and then I made a video recording for approximately 30 minutes or so. And then in a video editing program, I was speeding it up a lot. And then I could actually see the white blood cells move around. Yeah. So actually nothing unusual, I just basically let the camera record. Yeah. Let's have a look here again. Yeah. It would be actually nice to see. There is something that I am planning as well. It is indeed possible if you get everything properly set up, and if you add bacteria, you can actually see the white blood cells chase down bacteria. You can actually see that. Yeah. Yeah. But again, you have to speed it up quite a bit because it's they do move, but they move slowly. Yeah. So let's have a look here. Yeah, so these here look quite definitely like white blood cells. This one here, this one here. You can actually see in here, it looks that's the nucleus in here. Yeah? Not all white blood cells move, some will remain stationary. But because this is a non-stained sample, therefore they remain alive. If basically if you add a stain to it, you're killing them off, and then they don't move anymore. Yeah. But what one could do is one could look at one of them and see how it actually changes the shape and how it crawls along over time. Yeah. This one actually is not so good. I've actually seen all the better ones. Yeah. So I'm going to read why glycerol immersion is not more common. Is it much worse than oil? I guess it's still better than air. First of all, I know that there are companies out there. First, it's a question of refractive index. That's it, right? It's a question of refractive index. I know that there are some companies out there that are manufacturing immersion lenses, which are not for oil but for water. Okay, so that is called water immersion lenses. So you can basically simply dip the objective directly into a small tiny petri dish. So for example, you want to do so-called cell culture observations with the cells in a petri dish. You can use water immersion objectives where essentially you just have the petri dish and you just rotate the objective directly into the liquid of the petri dish. Yeah? So these things exist. Glycerol, I guess it has to do with the refractive index, and I'm quite sure that honestly, if you were to do research, maybe indeed there are people that have experimented with that. However, in science and research, what is really important is you want to have standardized, reproducible methods, because in sciences, reproducibility is so important. So for this reason, even if other methods probably would also work, scientists have agreed on certain common methods that everyone uses so that you can compare the results. That's simply something that I think might also be a factor here. Yeah. Can I add bacteria to those blood cells? I need to grow them first, but theoretically that is possible. And I'm currently growing some E. coli bacteria. It's really important if you're doing any experiments at home, you are not allowed to, it's not one that I don't allow you, but there are laws, you are not allowed to grow bacteria that essentially are unknown. You can only grow so-called biosafety level 1 bacteria, which are basically known to be harmless. So for example, bacteria that you have on food, cheese, for example, yogurt bacteria, these are biosafety level 1 bacteria. They're known and you know that they're not dangerous. But any unknown bacteria are actually, you're not allowed to cultivate them. Yeah? Look, this might be, let's have a look here. What we see here in the middle here, this might be a white blood cell. Okay? And I do have the impression that it seems to be slowly moving into that direction. Yeah, it's changing a little bit the shape. Yeah, and you see here the back part over here, right? It's kind of it's moving a little bit upwards like this, very slowly. So that is basically a white blood cell that's moving around. Yeah, and that's why you have to take a video, I don't know, maybe half an hour, and then you have to speed it up 100 times, and then you can actually see it crawling around the slide. Yeah, yeah. So yeah. I was hoping for that question here. Can't E. coli bacteria cause infections, so it does in a way cause harm? No, that's a very good point. Okay, the thing is that E. coli bacteria, you've heard from the media, newspaper and so on. Oh, we found some E. coli bacteria in drinking water. Dangerous, right? You don't want that. Correct. However, it's not the fact that they're E. coli bacteria, but the fact it depends if the E. coli bacteria carry DNA that actually makes it dangerous or not. I'm going to use a very stupid example. No, it's a bad example. How do you do that? E. coli doesn't say anything. Look, you cannot say that dogs are dangerous just because they're dogs, right? Dogs are dangerous if they bite. Right? And it's the same with E. coli. Not all E. coli bite, under quotation marks. In other words, not all E. coli bacteria produce toxins, poisons that are dangerous. Yeah, so that's a little thing. However, if you find E. coli bacteria in drinking water, they should not be there. Generally not. Whether they're good or bad, they should not be there because they're a sign of sewage contamination. Right? Look what happened to the white blood cell. It's now up here. Yeah. Yeah, so yeah, you are using DIC with 100 times. Okay, thank you very much. Ever Smaller is commenting on that. The whole issue with magnification is complicated because there are different ways of magnifying it, right? So there is one way of magnifying it is how many times bigger is it compared to the original size. The other way of magnification is this. Thank you very much. Here are a whole bunch of questions again, right? Other magnification is this, how basically how much closer do you see it and how much bigger is it compared to your vision. Then there's another type of magnification, how much bigger is it compared to the actual size. You know, you get the idea. It's a complicated thing. It depends also on a little bit the enlargement factor of the camera and so on. But I think what I wanted to show you here, I'm able to show you that this is a white blood cell, and you see it's been moving around, right? Yeah. And when we wait a few minutes again, then we can actually see that it's it's called phagocytes. So they're kind of moving around and they're trying to even eat stuff. And what I'm going to do next is the following. I'm going to do something, I'm going to add, after we watch this for a couple of minutes, I'm going to add a little bit of salt water. And then the red blood cells should actually shrink. Okay. Yeah, exactly. Ever Smaller just mentioned this. Yeah, I do want to add salt water. Actually, I prepared it here. Here it is. And this actually should cause the cells to plasmolyze. Yeah. The best way to test this is to look at a micrometer grid. Doesn't make sense to try to calculate the magnification. Thank you for the comment. No, I can't do that because I'm using oil immersion right now. But I do have a micrometer slide here. Look, I'm just going to very quickly change again to the desk view so that you see what a micrometer slide is. Okay, here, let's put the blood slide away. Do you here, maybe, I don't know. I'm going to hold it like this. This is a special slide, and there is a tiny microscopic grid on here, so that allows you to measure out the actual size. Yeah, you cannot see it. There is a really tiny grid on here, and then you can actually see the grid also under the microscope. I cannot show this to you right now because there is oil on my slide, right? But using this, you can actually measure out the proper the size, and that's by far the best way. Forget all of the calculations; they're too inaccurate. Yeah. So here we are again. So here is again the white blood cell, and you know what? We play around a little bit with some colors here, maybe, or with this. This is an upright field. Okay. Again, I'm using my 100 times oil immersion objective. I cannot go back. I cannot go to a lower magnification now because then the objective is going to be covered in oil. Yeah. How do they make this black print on the micrometers? I have no idea. I don't know how they are actually able to print it that small. Seriously, I don't know. Maybe some laser engravement. I have no idea. I really don't know. because I was actually wondering myself, because I remember I tried to make something like this myself using a printer and printing it on an overhead foil, but the resolution of the printer is not good enough. So yeah, chemical deposition, or maybe some kind of lithography or photography method, maybe maybe it's etched away, or I have no idea. Yeah, but there must be some kind of a technique. So I'm going to now try the following. This is going to be a little scary right now, because I really have to make sure that the liquid goes beneath the cover glass. What mechanism makes the white cells move? Well, that is inside a cell that moves are protein fibers, actin and myosin, and those protein fibers, they are sliding next to each other. It requires energy, ATP, but they're protein fibers that slide and cause part of the cell to extend, and this is the part that moves forward. Yeah, so this is actually a mechanism that is on the big scale you see movement, but if you actually go into the cell, you actually have sliding protein fibers. Yeah, and the mechanism actually is similar to the way that our muscles are actually also have sliding protein fibers that cause it to contract. Basically, microtubules are again a little bit different, but essentially there are it's actin and myosin. Yeah, but it is correct that microtubules are also inside cells, and they are used as a structure to pull for movement of vesicles along those microtubules, right? So yes, indeed, yeah, you have actin and myosin, and also microtubules that extend. So, for example, when you've got microtubules that polymerize, it's tubulin, and then it becomes longer, then it can also push out one part. Yeah, and microtubules are also able to, in flagella, for example, they're also able to slide past each other, and then they are able to also have a movement. Yeah, so let's quickly do a little bit of how do you call this, salt water. So what I'm doing right now is I'm going to take a little bit of. I'm going to go back to the desk view simply so that you see what I'm doing here. And I hope that this actually works. So a small amount of salt water. Okay. And this is not a difficult part. I have to now try to put the salt, check what I'm doing here on the side of the cover glass. And I have to hope that capillary action is able to do its thing here. No, it's not. Because sometimes the blood dries. If not working, then I'm going to make a new blood sample. And I tell you why. The salt water is not drawn in. And the reason is because the blood already started to dry on the side and does not allow any water to go in. Okay, that's the problem. I'm pushing right now. Maybe I'm able to. I don't know. Let's have a look. You see, that's the reason here. Everything is already dried up here on the side. Yeah, here not. Here not. And here it starts to be also not. Here everything is already dried up a little bit, and salt water is not able to reach it, it appears. I'm gonna give it another try. A little bit more. Yeah, so nope, don't work. Means I have to do another. No, it's completely, the cover glass is completely stuck to the slide. Yeah. That's actually maybe also one of the reasons why it does not dry out so quickly, because as it dries out, the edges of the cover glass dry first, and then it remains liquid in the center. Okay, so this means what I'm going to do now is I'm going to do another one. This time with a little bit more blood. And I'm going to mix in some salt water then again. So this means, what time is it? What time is it? Yeah. Actually, I do have a list of questions here. but I answered some of them already. I'm not going to force myself to stop now. I'm just going to, because I actually want to do this now. Just a second. I'm going to, again, I don't know. Did any one of you count how often I already pricked myself today? So here we go. Yeah, desk view. You want me to mix swamp water and blood, and to see how this reacts. So let's see. Here, let's take a little more blood now. Here. So you now know how red blood cells look like, right? And now I'm going to add a little bit of salt water. So I'm going to do this here again, using this, and now on the other side, I'm going to be adding a little bit of salt water. Let's do it right here. Yes, and they're meeting here in the center. Let's have a look how the blood cells look there, okay? So I'm going to remove this one here. I have to clean the, and I'm not going to use oil immersion now. So let's use, and let's have a look here. And I need to change back to the scope view. So here it is. So that's again, so very dense as expected. So, and let's have a look if over here, exactly, that's what I wanted to show you. This is where the red blood cells have contacted the salt water. And we can already see something happening here. Let's go up with the magnification again a little bit. That's exactly what I wanted to show you. And they behave exactly the same way that I hoped that they would behave. Yeah. Look, how do they look like? They look all wrinkled up, right? Yeah. So 60 times, so no oil immersion, so it's a little bit smaller again. And the cells look all shriveled up and wrinkled up, and that's because they have lost water. Water moved out by osmosis and caused the cells to shrink, right? And that's why they look so irregular. Yeah. That's, the demonstration here. Yeah, there are granulated blood cells. Essentially, they have also, I'll show you some of them here. They look a little bit like star shapes as well. Yeah, and that's the reason is because when they lose the water, then the membrane is too large, and then the membrane has these it's kind of outfoldings. Yeah, that's essentially what happens. Why drinking salt water is not good. Of course, if you drink salt water, the blood does not become in direct contact with the salt water. It goes, of course, first into the intestine and so on. But this is basically what happens to cells. And we can see it quite nicely here in the red blood cells here. Yeah? Okay. Maybe a question relates to the bikers getting sick from mud splatters. Recent video. Yes, I just recently made a video. I get it. Yeah, I just recently made a video. Now that's with E. coli thing. I recently made a video where it happened repeatedly. People who were riding a bicycle in races, they got quite ill afterwards because they got splashed mud into their face, and also they were eating some of this mud. Not the mud was the problem, but there were basically animal waste, droppings and manure, you call this, right? Was washed on the road, and then basically was the people who were riding the bike got splattered into their face, and that's why they got ill. And they analyzed this and found, of course, also E. coli bacteria. Yeah, but the question is why is E. coli safe? And I'm telling you, the ones that I have, these are laboratory E. coli bacteria, and they can also be eaten even. And essentially, it's like this, that they're also even in selling E. coli bacteria, the safe ones, right, to regenerate the bacteria in your intestine. Yeah, yeah. So basically, I think you get the idea, right? That they look kind of shriveled up and wrinkled up. And now I know exactly what some of you are thinking. If there's salt water, can we not try this also with regular water? Okay, this is the next question, and I will tell you, yeah, but it will not look quite as interesting, because what's going to happen is that the red blood cells, they will swell up, they will pop, and then basically you do not see them anymore, because the contents of the cells runs out. And then there is then basically you cannot see anymore the inside from the outside, the difference, and then essentially this appear to be disappearing, right? So adding regular tap water causes the cells to swell up and pop, and then you don't see them anymore, right? So that's a little bit the problem when adding kind of how do you say regular tap water. Yeah. So I got very it looks very different. What happens when you've got an infusion and the salty water goes into our veins? How salty the salty water you use? The salt water that you can use to essentially get an infusion is, to my knowledge, about 0.65% salt. That is really important. If it's any much higher, then this is going to happen, what we have over here happening. And if it's lower, then the red blood cells will pop. I'm going to tell you something which is not so nice, but it actually fits the topic. When you get water into your lungs, when you drown, right? There is a difference between drowning in fresh water and in marine water, in salt water. Because what happens is when you drown in fresh water, the cells are going to pop, because the water will go over the lungs into your blood. The cells are going to swell up and pop, and that's basically the problem, right? Just saying, yeah. So I also have read somewhere that back in the day, during the Second World War, what they've done is they've also given, when there was not enough salt water available at the right concentration, they've also given apparently infusions using coconut milk. So basically they used coconuts and they took that liquid directly. It was clean, right? And it apparently had just the right salt concentration or mineral concentration to be used as for transfusions. Yeah, just saying. So lots of interesting things happen here. But again, this one is concentrated salt water, and I think you will see quite nicely that the red blood cells have lost their shape. Yeah. So this is, yeah. Please research this coconut milk directly into the vein. That's what I read somewhere, and apparently it worked because it had just the right concentration of salt, and it was actually clean, right? And so free of bacteria if it's fresh. So that is basically the thing here. So I'm just thinking a little bit. Let's have a look here. See, that is how it's very dense. Yeah. So salt did not reach that, but over here, this is where I added the salt water. Yeah. And the white blood cells, that's basically you'd have to look for those. Yeah. And they will not have survived the salt water treatment. Yeah. So what camera am I using? I'm using a DSLR camera, a Canon D90, directly connected to my, oops, that's why you're asking. Which camera? This one here is the camera for the stage. That's a simple, unfortunately a little bit too slow USB camera. But the one that I'm using here is actually a DSLR camera, which produces an HDMI stream, which I put then into the computer. Okay, folks, listen. I have got a whole list of questions like every week, which we didn't get to answer. But I have to tell you the following. I actually answered most of them already. So there's no point in actually looking at those. And I thought maybe it's, you know what? We still have 5 minutes. Just for the sake of completion, I'm going to add some tap water. Okay? I'm going to add some tap water and we're going to see how this works, right? Again, the tap water, as I mentioned before, you will see much less. Okay, so don't be disappointed. But just as I mentioned, just so that you can see the opposite effect. And then what we have is we have red blood cells. They're essentially, we call them ghosts. They're actually called like that, which are basically just empty, how do you say, empty bags of a cell membrane. And the contents is completely lost. And because they're just empty bags of a cell membrane, you don't really see them very well. Those you see quite well because the cells are actually still complete. They're not popped open. They're just shriveled up, right? But when you add tap water, they should swell up and then they pop, right? And we're going to have a look at this, right? So this means I need some tap water. I've got over here. This one I need to get some, again, a new fresh pipette. Okay. And here we go again. Okay. Here we go again. Where is the desk view? As before. There's nothing. I'm slowly getting used to this. I'm not as, how do you say, hesitant anymore. But I also do feel that it's more and more difficult actually to prick myself. Here we go. So, number one. A little more maybe. So here we go. Okay. Yeah, we need a cover glass again. And in this case now I'm going to add some pure tap water. Okay, so it's exactly the opposite effect that we had. So let's move this up here. So here we go. The blood spreads again, and on the other side I'm adding some tap water, and I hope that they will actually meet each other. You gotta be careful, there's some air bubble in the center. Yeah, but I think they've met each other now. I hope so. So let's check. Let's check. I don't know. Yep. Okay. Again, I'm going to have to remove this here. Where is the scope view? And here is the tap water. And so, yeah, here's the blood, right? And I'm trying to find again the so let me have a look. Yeah. Oh yeah. You see that here, they don't look shriveled up. Let's go up with the magnification a little bit. So let me see. Yep. Yes, that's actually, yeah. And what happens here is you see that they start to really fade out, right? They start to become brighter and brighter and brighter, and then they appear to be almost disappearing. Right? Yeah, you see they're very, very transparent now, and the reason is because of what we have here are they're so-called ghosts. They pop open, and the only thing what you have is this empty membrane. Yeah, and you see also that they're much rounder because they're kind of blowing up like a balloon, and then they pop open, and then they become the contents runs out, and then basically we can also see that the surrounding medium becomes a little bit more red as well as the hemoglobin spills out. Yeah, see over here, almost invisible already. Yeah, so these are not empty bags of membrane because by osmosis the cells popped open. Yeah, they almost appear to be disappearing. Yeah. So that's kind of the idea. So there is a question here by Marie. Sorry if I missed it, but how can we get venal blood instead of arterial so we can compare? Honestly, I don't know if there is going to be such a huge difference, because if you actually take blood from the veins. Right? Blood from the veins, which actually carries, in most cases, there are some exceptions, deoxygenated blood. As soon as you put it on the slide, it's going to be in contact with the oxygen of the air, right? And then it's going to absorb the oxygen. So I would not say that you would be able to see a difference, and the shape of the red blood cells should not change because of that either. Yeah. So this is the thing. A single drop of blood have life? Well, that's a philosophical question, which is very interesting. Let me talk about this as well, and then I would like to call it a day for today. So again, for those of you just joined or kind of missed a little bit, I added now, how do you call this, tap water, okay? And this tap water causes the red blood cells to swell up and pop open, and therefore we don't see it quite well. So I'll leave it up to you. I'm not saying that the red blood cells are alive or not alive, but I'm just telling you the following. The red blood cells do not have DNA. They do not have a nucleus, right? They lost the nucleus during the cell division process because all of the space is needed to carry hemoglobin, to carry oxygen. So no nucleus. The red blood cells cannot divide anymore, right? Yeah. So if they're not able to reproduce on their own, are they then alive? Yeah. They do not have a full metabolism either because they're just basically empty bags floating around with hemoglobin. Yeah. So should you call red blood cells now alive or not? That is a little bit a question of definition of debate, right? I'm just saying, I think it's interesting because you wouldn't call them dead either, right? I mean, but they're not really alive as other cells are alive because other cells, many of them at least have the possibility to divide. Yeah, I was just talking ever smaller, was just talking about this with my Discord friends who are watching along. We agree they aren't alive, they don't die, they get destroyed, they get broken down, indeed, right? And recycled. Yeah. Are viruses dead? No, they're not dead, but they're also not alive. You have to be careful. Not alive does not mean dead. Dead means that they were once alive. So, for example, when you've got, a plant that's dead, then you know that it was once alive. Viruses were never alive because they do not have their own metabolism. Yeah? So there's a difference between something being dead and something being not alive. So just to show you, anything, any object, a pen here. Yeah, the pen is not alive, but I would not call it dead. Right? It's simply not the right term. So that's why I'm saying it's a philosophical question, a little bit on word. What do the words mean, right? It's a language question, right? So this the thing a little bit here. Okay, so it's a little bit. Sometimes you see. Maybe this debate, what's life, what's not alive, and so on. Maybe this debate reminds me sometimes a little bit of the debate of should Pluto be considered a planet or not. You see, it's a question of what label do we attach. Yeah. Okay. When it runs out of blood or ink, is the pen dead? Then you know what? Because Maradona is saying, I think I'm having a heart attack because of this. I'll tell you the following. And then I'll just leave it with that today, just to stress you a little bit. Look, you know, this is my wristwatch, right? Living things move. So does my wristwatch. Living things respond to stimuli. So does my wristwatch. Living things use energy. So does my wristwatch. Is my wristwatch half alive? I'm asking. Yeah. So this is just something that as a matter of fact, the question of what's life is a very difficult question. And we're seeing this right now because NASA just a few days ago has announced something that they found some structures on Mars that they cannot explain. They think could this be life or not? You see, it's a very interesting but difficult question. Yeah. The watch is alive, okay? Yeah. You see a difference in your blood when you're drunk, like 2 per 1,000. I probably don't think so that you would see a big difference when you with alcohol visibly. However, one thing where you can see a difference, it's not seeing it, but actually the blood. If you're very thirsty and if you have not been drinking a lot, then the blood indeed is thicker. And just as a little interesting story, once remember when I told you that the doctor tried to get some blood out of my earlobe for testing, and they couldn't get any blood out, right? It simply wouldn't flow. And then the doctor says, well, you have not been drinking enough. So I had to drink a lot. I had to wait a little bit. And then basically the blood started running better because there was also more water in my blood. So it's not a visible difference, but actually there is indeed a difference in the way that the blood behaves. Yeah. So yeah. So ladies and gentlemen, I think I'm gonna call it a day today. Yeah, this was for those of you who basically joined in late and who wanted to see the beginning. The video will be, of course, available online as well. I hope you found it interesting. Okay, I've never been pricking myself so often. Yeah, that's the 3D printed red blood cell, right? Like today. And now I have to kind of clean up the mess a little bit here. And I'm going to be thinking again of a topic for next week. Yeah, I'm always grateful for any recommendations that you might have. So I have already an idea. So maybe I'm going to put some bacteria next week under the microscope. I have to see if I'm able to grow some of them. I wish you all the best, folks. Okay, happy microbe hunting as always, and all the best. See you again. Bye bye.